Hello, everyone, and thank you for joining this installment of Boyce Thompson Institute's new Breaking Ground discussion series. My name is AJ Bushy, and I'm in the Communications Department here at BI. And today we will be in conversation with Drs. Fay and Giovannoni, who are both professors at Boyce Thompson Institute. And today they will be telling us about how they created something called the tomato pan-genome and how it can be used to improve tomatoes. Hi, Jim and Fay.
Hello.
Before we get started, I would like to take a moment to thank today's program sponsor, Sullivan Trading Company, a reliable source for canned steamers and repairs for the canned food industry for more than 25 years. You can find them on the web at sullivantrading.com. And a few housekeeping announcements. During the session, all participants will be muted. You can ask questions in the chat and in the little Q&A box down below, and then we'll do a Q&A session at the end of the conversation. So thank you all again for joining today. And let's start with you, Fay. Could you tell us a little bit about yourself and your lab?
Thank you, AJ. So I grew up in a very rural, okay, great, thanks. Thanks for sharing the slides. So I grew up in a very rural village in Anhui province of China. Sorry. So actually, I'm the first person in my village who went to college. Wow. I got my bachelor's and a master's degree in horticulture from China Agricultural University. And I did my PhD study actually in Jim's lab and obtained my PhD in Texas A&M. I then worked at Virginia Bioinformatics Institute for 2 years before joining BTI and USDA in 2006. So time flies. I'm here for almost 14 years.
Wow.
Thank you.
Yeah. And so what is your, what's your current focus? You do bioinformatics type research, right? Can you tell us a bit about that?
Showing on this slide, so my lab is purely computational, and the projects in my lab are highly diverse and highly collaborative. And the funds supporting the work in my lab are mainly from National Science Foundation, USDA, and the Gates Foundation. So my lab, the one focus area in my lab is using integrated bioinformatics and the genomics approach for like trait discovery, crop improvement, and then knowledge advancement. And my lab also developed computational tools and a biological database to efficiently store, disseminate, and analyze and integrate large-scale genomics data.
So you both mine the data to make discoveries as well as kind of keep it all in a nice database and create tools so other people can also...
Yeah, other people can use it, it's a user-friendly interface. So, yeah. So people who don't have programming experience, they can use the data more efficiently.
Very cool. And obviously the COVID pandemic is affecting us all right now. How are you in your lab? How are you coping?
Ooh. So at the computational lab, the impact of the COVID-19 to our research project is not as much as for other labs at the BTI, the wet lab at the BTI. As we can still work from home on these projects. But I have to tell you, it's not fun at all. And the last time I saw my lab members face to face is in March, before the lockdown. I haven't met them for like 4 months. So and the BTI policy, so my lab members was not allowed to enter the building for work because my lab is computational. We can work at home. And the telecommunication to discuss projects sometimes works, but it is not that efficient as run by to the office close by, talk to in person. And but I kind a little bit frustrating. But anyway, a good news is that most of the projects in the lab are on track. thanks to the hard work of my current lab members during this difficult time. And I appreciate it, really appreciate it.
Yeah, that's good to hear. As you know, we've all been living with a lot of working at home, and it can be tough. So it's good to hear that your lab members are.
Yeah, actually, this picture was taken last year in 2019, in the summer of last year. They had summer interns, so you can see last year I have a lot of people, but now I only have 5 postdocs and 1 visiting scientist in the lab. There's a lot of people leaving, and also I have the biggest impact of the COVID-19 to my lab is that I have 2 new hires in China. They can't come to join to my lab due to this travel bans, the pandemic.
Yeah, we can just cross our fingers and hopefully this will end soon.
Hope this will end soon.
Yeah, exactly. Okay, Jim, would you mind telling us a little bit about yourself?
Sure. So thanks, AJ. Thanks everybody who's joining in. My name's Jim Giovannoni. I grew up in San Francisco, California. Went to school at UC Davis, and then got my PhD at UC Berkeley. Came out to Cornell for a postdoc in the early 90s. Had my first faculty position at Texas A&M University in the Department of Horticulture. That's where I met Faye. And then came here in 2000 and have been housed in BTI since then, so just about 20 years. I'm also actually a USDA ARS scientist, so I work for the feds, and I'm director of the USDA lab, which is just across the street from the BTI, for some physical perspective.
And so what does your current research focus on? Tomatoes?
Yeah, we do a lot of our work on tomato. We do some on melon. We work on other species as well, to the extent that they help us ask questions about fruit ripening and fruit development and the nutritional quality of fruits, and particularly some of the biochemical pathways that contribute to nutritional quality. So a lot of our work in other species is focused on asking whether do things we learn in tomato apply across a broader spectrum of diversity.
And there are lots of species in the same family, right? In the family?
Yeah, I mean, so in the Solanaceae, which tomato is a member of, are a lot of the really most important vegetable crops. So related are potato, eggplant, pepper. Kind of the dark sheep of the family is tobacco, which is a very good experimental system for asking questions about plant biology. And then some of the more distantly related but not so far away species include, for example, coffee, which we do a little bit of work on, just asking to what degree fruit development in coffee has some similarities to tomato.
And how is your lab dealing with the pandemic? It looks from these pictures that you're all masked up and.
Yeah. So you can see, I mean, I guess the good thing is we're still getting some work done. Everybody's masking and social distancing, and so it certainly has had an impact. We can't have all the people in the lab that we normally would. We're not a huge lab, so we're able to get some things done, but people are kind of rotating in and out to minimize our footprint at any given time. Right now the lab's 9 people. A couple of those are mostly working at home. One's working in China because he went to Wuhan to visit family the week before all this happened. So he's kind of stuck there, but fortunately he's working in a collaborator's lab, so keeping things going.
That's great. Okay, so thank you for those introductions. And so I guess, so the topic of today is the tomato pan-genome, which I'm guessing a lot of people haven't heard of. It's kind of a new thing. Jim, could you give us a little background, kind of what led up to the pan-genomes and the history of the tomato and tomato genomics?
Sure. Just kind of from a very general perspective, tomato has long been one of the species that experimental scientists use to ask questions about basic plant biology, flower development, disease resistance, and there are people should go look at the BTI website because there are very excellent scientists here at BTI that are also working on tomato that are not part of today's discussion, but are doing really interesting things. And it's because of its importance as a crop, has been a species that a lot of geneticists and breeders have worked with, and for example, have found a lot of mutations, just natural spontaneous mutations, which create a great collection or resource for doing experimental biology. Relative to what's shown on the slide that you're looking at here, if you follow this kind of convoluted tomato plant back to the little yellow star, you get a sense of the origin, the location of origin of wild tomato species. So they're typically found in northwestern South America, places like Peru. You can find them on the Galapagos Islands. And what's really interesting is they inhabit a range from literally species that grow right at the edge of the sea. So, and think about that in terms of what that means of the genetics of the plant. So you've got species that are very resistant to salt. You have others that grow in the Andes right at the tree line, where in some cases the wild tomatoes are the highest elevation species growing, so they can deal with high UV light, drought, cold. So there's a lot of adaptation in tomato, which again makes it a really interesting system to work with. And of course, groups like ours are interested because of the fruit, which are very important. Tomato was probably domesticated or was domesticated by Native Americans in probably the region around where it originally evolved. But the Aztecs are kind of credited with moving tomato, the cultivated tomato, the domesticated tomato, through Central America, and possibly Columbus, and certainly Hernando Cortez, the explorers from Europe, were the first to bring tomato back to Europe. And I think what's interesting in this slide you have here, this image from Mattioli in the 1500s of essentially some of these tomatoes, which he described as kind of weird eggplants, because they knew eggplant, they didn't know what tomato was at that time, were pretty similar to what you see now. And nothing like the wild tomato. So domestication had already occurred in the New World, and again had been brought up into Central America, and then brought back to Europe. Early tomatoes were probably consumed, but also a lot of people were afraid of them because they look a lot like nightshades that people knew were poisonous. Particularly the bittersweet nightshade has a red fruit that looks a lot like a cherry tomato. And the plant itself looks like the deadly nightshade, belladonna, although the fruit are quite different in terms of color. But there's a lot of probably concern about whether they could be eaten. In the US, Thomas Jefferson's often credited with making them popular. They were certainly grown. There's records of them being grown in the Carolinas in the early 1700s, but Jefferson was kind of a foodie. He had probably eaten them in France when he was ambassador there, and grew them extensively. And in his notes of his garden, you see a lot of references to tomato that probably were some of the ones he may have brought back from France, and others that were already being grown in the US. And so really in kind of the 1700s and onwards, you see a lot of reference to cooking tomato, eating tomato. And then, a lot of breeding has been done really after the World War II, a lot of modern breeding, but still using some of the main material that was brought back by the early explorers. So it wasn't until later, more recently, that geneticists have gone back to that wild germplasm and tried to bring in some of the interesting traits, particularly things like disease resistance, but there's a lot of other opportunities. And many labs, including our own, are interested in that diversity. And that led, first to sequencing of the tomato genome, which just captures a single genome. In that case, it was a variety developed by Heinz. The main reason that variety was chosen was because somebody had a big bag of seed of it. So there was no other real reason to select that particular variety. But because genomes of a species are typically very similar individual to individual, it served as a good reference. But a lot was still missing. And I think Faye can probably tell you more about that.
So so let's move on to Faye. Can you talk a little bit about what may have been missing from that Heinz genome and what led to the creation of the pan-genome, and what is the pan-genome?
Yeah, thanks, AJ. So first, I may give you some background on the pan-genomes. So the concept of pan-genome was first developed in bacteria in 2005, and then later applied to human and animals, then recently to plants. So a pan-genome should contain the complete or all genetic elements in a list of organisms. And the pan-genome can include a core genome and accessory genome. The accessory genome also called variable and dispensable genome. So the core genome include genetic elements shared by all organisms, while the accessory genome includes genetic elements shared by a subset of organisms. So actually, the accessory genome is that's the reason causing the diversity of the organism in a species. So as shown on this figure, you can clearly see the phenotypic diversity, and later there's a report on a very high metabolite diversity in tomato between tomato cultivars and also in the wild relatives. And you can see on the right bottom, that's the pimpidifolium, this small currant tomato. That's treated as the progenitor of cultivar tomato. And then we have this hepatitis. You can see the trichome, the right top corner, that green one. And then you can see some tomatoes, they can grow, the wild tomatoes can grow in the very hard conditions. And this all controlled by some unique genes to adapt those conditions to contribute to those unique phenotypes. So that means that there must be some genes that are not exist in Heinz, in the reference genome. So the one reference genome is not good enough for tomato now. So we realize that we need to start to capture the genetic element from all these different individuals and make a complete catalog of the genetic elements. That's the tomato pan-genome. That's what we wanted. That's what we did.
So if I could just pop in a little bit, I think it's important to kind of define what a reference genome is. So we have this Heinz genome. And so when people might look at the wild progenitor and look for genes, but they would just compare it against that reference genome, right? They were looking at the genes that they share to one another. They weren't looking for new genes in the wild, unknown genes, I should say, in the wild progenitor, correct?
Novel genes.
Right, novel genes, yeah. So that's kind of what the pan-genome does, is it looks at everything from all these different strains and species. And
I think one big problem when using the one single reference genome is that our collaborator has this kind of experience. So they are interested in one gene, but that gene was not present in that reference. So they can't use that reference to study that gene. So that's why we're trying to build a pan-genome. And as a first step, we are focusing on the lycopodium clade. So that's the red clade of tomato. That includes the pimpidifolium that's a current tomato. And then the ceratiforme, that's a landrace, also called the cherry tomato.
So that's everything on this slide? That includes everything on this slide?
So this one, the bottom right, that's the pimpelifolium.
Yeah.
Yeah, that's a current tomato. Then the bottom left, that's a cultivated tomato, the big-sized tomato. That's a modern tomato.
Gotcha.
I'm not sure whether we have cherry tomato here. I think we have some cherry tomato on the top left.
Top left, looks like there might be some in there.
Yeah. And also we also in that red clay, like Poskin clay, we also have some very close related wild relatives like Chismania, Galacbense. So during the past several years, like 5 or 6 years, in this 5 or 6 years, people started to generate a lot of sequencing on those accessions. But those they are not in reference level. They are trying to generate the kind of like we call it deep resequencing of each accession, and then align those sequences to the reference genome. They identify single nucleotide polymorphism, and they already provide quite a lot of insight in the tomato domestication, the diversity, and the population structure. But still, they're also missing a lot of information that are not captured by the reference genome.
So they could find new versions of genes that were in that Heinz tomato, but they weren't in novel genes.
So that's what we collect all those data. It's about the genome reconstruction together of about 725 tomato accessions in this clay, in this red clay. And we built a pan-genome. The approach we use to build the pan-genome is called map to pan, map to the pan strategy. So we de novo assembled for each accession, we de novo assembled the short reads and generate contiguous. And then we map that contiguous to the reference genome. And for those that could not be mapped, that's kind of like novel genes or novel sequence.
Those are the novel pieces, right?
Yeah, novel pieces for each accession. Then we combine all the novel sequences from all these accessions because most of them are redundant. So we remove redundancy, we got a unique set of sequences that are not present in the reference genome. So that part we call non-reference genome, but non-reference sequence. You can also call it novel sequence. Yeah. And in total, we identified about 5,000 novel genes that are not present in the non-reference genome. There's of this 5,000, there's a very small portion. It's about a couple hundred of genes that are actually in hands, but they are not assembled in the genome. That's normal. That
so they're missing. They're in the Heinz tomato, but they were just missing from the.
Yeah, they're just missing in the reference genome because any reference genome you can't capture 100% of the genome.
Okay.
There must be some missing, So that's 200 genes. It's less than 1% of the genes in the tomato genome. The tomato genome has about 35,000 genes, and among this, a couple hundred genes that are not present in the reference genome, but in the reference accession. They include a very notable one. It's called GAM8. It's a glycoalkaloid metabolism 8 gene. So this gene involved in the regulation of glycoalkaloid biosynthesis. The glycoalkaloid is also contains the tomatin. Yeah, that's a very important chemical that involved in insect resistance and other health benefits.
So it's kind of like a natural insecticide, kind of?
Yeah.
So,
And we also identified quite a few genes that are not present in the reference genome and also not present in the reference. The notable one is the PTO gene that cloned by Greg Martin's group, involved in disease resistance. And also the I2C1 and HCR9, those are genes, the very notable genes that are involved in disease resistance that are not present in the Hansa accession.
so it sounds like there were a lot of genes that would be pretty useful for breeders and farmers that were missing from the Heinz genome, but they're present in the wild and some of the other wilder relatives.
Yes, exactly. And also that people realize the importance of wild relatives. And they know that wild relatives contain genes that are absent in cultivar tomato. And now the wild relatives have been commonly used as a source for beneficial traits. Mainly disease resistance, the integration, integrate the disease resistant genes into cultivar tomato to improve the particle stress resistance.
So how does the pan-genome make it easier to find those? Is it just that there's this database where everything exists and everybody can just kind of go in and look for stuff, or is there something else more than that?
Yeah, the sequence, the novel sequence and the genes, they are public. Yeah, they're public.
Okay. So speaking of finding new genes or new versions of genes, you guys found one, right?
Yeah.
Jim, you want to talk about that one?
Sure. So let me first say one of the pleasures of having students and postdocs in your lab is that you have the experience of having somebody come out who's a better scientist than you are. So that's certainly the case with Dr. Fay here. And he actually found this gene. Well, kind of the interesting story of this was, he said, here's some genes that when we look at this collection of 700 plus that we've sequenced that are found in very few, so kind of rare genes. And it turned out that, so this particular gene is called TOMLOC-C, and it's involved, had been discovered previously, a different version of it, and shown to be involved in lipid metabolism or fatty acid metabolism. And breakdown of lipids can result in volatile compounds that influence aroma, and aroma influences your perception of flavor. I think a lot of people know this, but if you have something very aromatic, you can kind of demonstrate this at home with a nice wine or a strong cheese. Just hold your nose while you're drinking or eating it, and you'll get a different flavor sensation. And so some of these volatiles are important in how we perceive flavor of the foods we eat, including fruits, including tomato. And what was interesting about this gene is it's a version that's more highly expressed in the fruit. And simultaneously, what was going on in our lab was we were trying to map regions of the genome that influenced flavor and also regions that influenced color. And it turned out we had a region that seemed to influence both, and this particular gene sat right there. So that made it extra interesting to us because we thought it might actually be the gene underlying differences in color and aroma / flavor. And it turned out that in addition to fatty acids being different when you have this gene, so were carotenoids. And carotenoids are a class of compounds that are in tomato related to the colors of the fruit. So lycopene is the primary carotenoid of tomato. It's the reason they're red. Beta-carotene is the reason carrots are orange. And actually in the biochemical pathway, lycopene is converted to beta-carotene. Downstream of that, it's broken down into compounds that you can perceive as volatiles. That is, they're volatilized, they go into the air, you can smell them. And an example of one called beta-ionones shown in this slide, and that's actually one of the volatiles that when you do taste panels, people associate levels of that with, oh, that's a good tasting tomato. And so what was interesting was this gene seemed to be responsible for both fatty acids and carotenoids being broken down, and they're different chemicals. You can see that if you look at these images of linoleic acid and beta carotene, but somehow the enzymes seem to be influencing both, and probably what it's doing is it's actually breaking down fatty acids, but some of those intermediates are interacting with carotenoids and resulting in changes in the aroma as a result. So what was interesting when we looked at phase data was where is this gene in kind of the collection, and it's in the wild species. It's in accessions of Pimpinella folium, which, as Faye mentioned, is the wild progenitor of the cultivated tomato. It's the only red-fruited wild tomato species, and then it seems to have been lost. It's in almost none of the kind of older varieties. And what's really interesting is you see a little bit of a resurgence in modern varieties. And why that's interesting is probably because it's really just in the last 20 years or so that breeders have started focusing more on flavor and other kind of quality traits. In the past, breeders, essentially growers were paid, and therefore breeders produced fruit that were big and red and at high yield. So they had to perform well in the field, and you had to fill the box with big red things. But people weren't really tasting them a whole lot, and I think a lot of consumers realized that flavor is not always optimal, especially in store-bought tomatoes. But what was interesting is we see this gene has been coming back. It's a little more prevalent in more recent varieties, probably because in the last 20 years or so, breeders have focused a little more on flavor and other quality traits. And I think, people who've been around long enough remember that you'd go to the supermarket and you'd see 2 or 3 varieties of tomato, a cherry, maybe something kind of in between, and then a big beefsteak. Then after a while, the kind of Roma varieties showed up, the kind of elongated ones. And now you see all kinds of colors and shapes and sizes, and that really reflects the breeders recognizing, and the seed companies and the growers recognizing that consumers want something that has a higher quality. So we believe this gene is one of the genes that was maybe lost, probably inadvertently, just because people weren't selecting for it. Might have been linked to something else bad, so it might have been selected against. But it does seem to be making a resurgence. And now with the pan-genome and the availability of the gene, breeders can actually target it and try and bring it back into modern varieties.
And we did find, or I think you found a list of some varieties that have this version of the Tom-Lock-C gene, right?
Yeah, when we look through the ones that were sequenced, there's a handful that have had it, in addition to the Solanum Pimpinella folium. So I made a little list. I wrote it down because I can't remember, but people always ask. So Gardener's Delight is one. These are all varieties that most of them you can find in online seed stores, particularly the ones that emphasize kind of heirloom or older varieties. Super Sweet 100, Marcelino, Galina, Katinka cherry, which is a Russian variety, I believe. That's probably a little harder to find. Yellow cherry. So some of these are kind of yellow, white colored fruit. And most of them will breed true. The Super Sweet 100 and the Marcelino are hybrids. So those if you save the seed, you won't be able to regenerate the same plant back, but the others you will. So they're so-called inbreds. But in that 750, that was kind of the list. There might have been a couple more I missed, but there was a dozen or so that had it, and most didn't.
Very neat. It's a really neat story about how good things can be lost over time. when you're breeding for one thing like size or shape, flavor and taste, and like insect resistance, drought resistance, all those kinds of things. So
yeah, I think the pan-genome and what Faye discovered showed this is one of 5,000 opportunities, right? So this is one that was pretty well characterized, but there's many others, and just looking at their sequences indicates, for example, a lot of them look like they could be related to pathogen resistance. And again, if in the case, this is a case where the gene was probably lost. In many other cases, it just was never in the original progenitors of tomato. It wasn't, it's in some of the other wild species which can be crossed, but are not always used in trying to improve tomato. So knowing they're there makes it a lot easier to try and deploy them.
Well, it's fascinating. It's a great resource you've created, and I'm sure researchers like Jim and you, Faye, will find lots of neat new stuff that will benefit us all who like tomatoes. Those who don't, because they don't taste good, but they will taste better. Okay, let's go to some Q&A. We have a couple questions in the chat box, and again, if anybody wants to ask a question, feel free to put it in the chat box or in the Q&A box. So we have one. The first is there evidence that native peoples intentionally bred these more ancestral tomato forms? I'm guessing like in Mexico and in the northern Andes.
Yeah, I think it's clear, as I had mentioned earlier, that what had been done by the Native Americans was essentially creating the modern tomato. Right. So what was brought back to Europe was essentially very similar to what we have now in terms of sizes and shape. There's been a lot of breeding since to add additional characteristics, but it's pretty clear that the original domestication, the original development of a kind of a consumer-oriented tomato was done in the Americas by the native peoples there long before the European explorers ever made their way over. What they brought back was, you can think of as a pretty early finished product that since has been undergoing a lot of improvement. And what's interesting now, I think, as we've said, is now breeders have better resources, better tools to go back and reuse or try to use some of these wild species. From a breeder's perspective, it was always difficult to take the image that Faye showed of, for example, this green fruited tomato looks nothing like what you get in the supermarket now. And it might have a very useful trait. There might be a resistance to an important disease, but as soon as you cross that, you create something that you then have to spend years and years bringing back to what is similar to a modern tomato. Now there's a lot of genetic resources, a lot of populations where a lot of that work's already been done. It makes it a lot easier for a breeder to kind of start with something that might have that wild species gene, but looks a lot more like a modern tomato to begin with. So a lot of breeding now is using these types of resources and essentially working with these these older species or unused species, and can take advantage of now what Faye has found in terms of the pan-genome, describing what genes are there that hadn't been identified in the initial genome sequence.
So it'd be much faster now to, presumably you could cross these two, like you mentioned, and then just take the seeds and just check it out, see what genes they have, see if they have the genes that you want, rather than having to grow them and, and keep doing a bunch of more tests. Is that?
Well, yeah, I think, there are types of populations. An example is something called an introgression population, where someone has taken a wild species, crossed it to the cultivated tomato, and then done many back crosses with the cultivated tomato, and essentially creates a population where each individual line has a little piece of that wild species genome. And so now the plant itself is much more like a cultivated tomato. But if you're interested in a trait that's on that little piece, you can use that rather than the whole wild genome,
Right.
Which will have a lot of things you don't want, right? You don't want this little tiny fruit. You don't want green fruit unless you're making fried green tomatoes. There's a lot that has been done through domestication and breeding that's in modern varieties that are pretty essential. You want to bring in the tweaks, the resistance to a particular pathogen, the changes in flavor, maybe optimization of color. And then, the reality is there is no perfect tomato, because culturally and individually we like different things. And so there's a lot of perfect tomatoes for a lot of different people. So that's the job of breeders, is to meet those different subsets of kind of the broader market.
That's a good point. That probably varies geographically as well, I'd assume.
Absolutely, yeah.
Some people may have a sweeter, more of a sweet tooth. Others do not.
Sure, yep. In the US, we tend to like sweet things, and the varieties here people prefer tend to be sweeter, a little more acidic. When you get into Asia or into Europe, some of the selections in Asia early on had color variation that now is kind of what's considered the normal, and for us might look a little pale. But so yeah, people have different expectations, preferences, and the job of breeders is, how do I maximize what they're looking for plus add what's going to make it better to the consumer. And again, this is a resource to do that.
Yeah. Okay. Now let's move on to the next question. For tomato, this sounds like one for Faye. A tomato that is about 950 megabases. How much deep short read sequence did you do, single or pair ends?
Yeah, I suggest always using paired end. For genome resequencing, always use a paired end.
Okay. That
helps mapping.
Okay. I'm not entirely sure what that means, but is there a reason? Could you share why paired ends are better?
Paired ends, that means one molecular sequence at both ends.
I see.
That means the uniqueness of the molecular will be double. So that helps mapping to the right position of the reference genome.
That makes sense. So you're getting it both directions.
Yeah.
Just one, and it may end at some point. You don't know. Okay. Makes sense. One more question.
No, I think she has an additional question. What's the depth, right?
How does the sequence variation within domesticated tomato varieties look
AJ,
like? What?
I think Pat has an additional question, the previous one. Not only single end or paired end.
Oh.
And I will receive the avocado wild accessions, and I would like to have your suggestion on how deep I can do. I try to answer this question. It's a very good question. So for tomato, because most tomato are inbred line, they are highly homozygous.
Mm.
So the depth is okay. If in low depth, you still get a very good assembly. And for avocado, it's highly heterozygous, and that could be different from tomato. And actually, we have experience in apple. So apple is also highly heterozygous. We did the analysis we randomly select different depths of apple resequencing data to see what kind of depth is optimal, cost-wise, and also give you the enough information. And for apple, we tried from 10x, 20x, till 100x. We found at 40x, they are good enough. The 40x coverage is good enough, 40 or 50x. Then if you increase the depth, like 100x compared to 40x, there's no much difference. So I'm talking about the de novo genome assembly. If your purpose is to identify SNPs, that's different. I mean, SNPs, I think 15x coverage is 15 to 20x coverage is good enough. Okay. Thank you.
Would those numbers vary based on size of genome or how many genes are in it, or it's pretty fixed based on?
No, it's not based on the size of the genome. It's based on the heterozygosity and also the complexity of the genome. Like if the genome has a lot of repeats, then the coverage could be more. I highly suggest maybe you can sequence in one accession first, like to do like a deep sequencing, like 100x or even 200x coverage. Then do a pilot study to check what is the optimal depth for your study, because this varies across different species, because their genome is highly different.
That makes sense. Okay, next question. How does the sequence variation within domesticated tomato varieties look like compared to the sequence difference between Pimpinellifolium and the reference genome in terms of percentage of sequence similarity?
So the domestic tomato, they have very narrow genetic diversity. So their variation is very, very small. But the Pimpinellifolium genome and the reference genome, their difference is not that huge. I think that in the tomato genome paper, we report about, I think, 6 million SNPs were identified between the Pimpinellifolium genome, and then the Hans genome. That's 6 million out of 950 million. It's less than 1%. So the sequence, most of the genes, if they're present in both genomes, they are largely identical. But they also have a lot of variations. So actually, we sequenced one Pimpinellifolium genome recently. We found a huge inversions. That's more than 10 million base pair long in size. The inversions between these two genomes. And we also found a lot of big deletions and insertions. Some are even megabase scale. Wow. We found a deletion that I think is 1.6 million base pairs.
Quite a bit different.
Yeah, quite a bit different. But in terms of the sequence similarity, they are largely identical.
So I guess now's a good time to mention. I don't think we did this earlier, but this tomato pan-genome was published in Nature Genetics last year. So folks want to read some more details about some of these methods and some of the other discoveries, you can find that there. And I don't see any, oh wait, there's some in the Q&A box. So we've got about maybe like 5 more minutes. We'll try to get through as many questions as we can. Jim, could you talk a little bit more about TOMLOC-C? When you say this gene is coming back, does that mean it is a wild type specific gene? And why is it coming back?
Sure. Yeah, all I meant is that the prevalence of the gene in more modern varieties has increased relative to older varieties. So what that means is that breeders are selecting for it, right? Whether it's, they're probably, up until now, they're not doing it intentionally. They're doing it based on the phenotype, presumably, of flavor. So it's, if you are tasting your tomatoes and you've got something that's helpful for flavor, you're probably going to be selecting for genes like this. If you're not tasting for flavor, if you're not testing for flavor, if you're not tasting them, you're just asking, are they, big and look good, which is also important, then a gene like this might get lost. Essentially, there's no pressure to keep it. So over time, it disappears. So I hope that answers the question. So what I'm saying, it's coming back. It just seems that it appears that breeders are selecting for it in more recent breeding attempts.
So breeders may be putting more focus on flavor rather than just size and shape.
Exactly, which we know they're doing. Yeah. We know that's happening in the last couple of decades.
Maybe I can clarify a little bit. So the TOMLOC-C gene is actually the presence in both wild and cultivated tomato, but there's a allele, the deletion insertion in the promoter region that controls the TOMLOC3 expression was lost during the domestication.
Yeah,
and that allele was increased in modern tomato variety.
Yeah, I think, right. So thanks, Faye. That helped. I think you're right. If the person was asking, well, isn't this gene there to begin with? The answer is yes. The version we're talking about is a different version of that gene, and that's what's been selected.
Good.
That clarifies, I think, a lot.
And how do you narrow down to a few candidate genes? I'm not sure.
Okay, I think, well, do you want to do this, Faye? I can try.
I think we combine all kinds of different information, right? And try to find something that are interesting and also important. And we have quite a lot of candidate genes, but you can't do all of them. If Ancestral give me the funding, I can do it. But we need a focus. That's why we select this gene. And also there's ongoing project in Jim's lab focus on this area. And I think it's a perfect combination between pan-genome and the experiment done in Jim's lab.
So you found one and just decided to look at it, basically.
Yeah.
Sounds like.
Well, yeah, as I mentioned, we're coming at this gene from a different angle. Right? In the case of TOMLOC. But
okay.
You know, the more general question of how do you pick candidates, it's really a question of what are you interested in? You know, what traits or characteristics are you focused on? Or is there some other, you know, what is your driver for your investigation? That presumably will narrow what types of genes you might be looking at.
Okay, this is going to have to be the last question. In addition to ancestral gene sequences, there appears to be a surprising amount of expression heterogeneity of a single gene in a single species in the same tissue or over time. By expressing heterogeneity, I'm referring to the observation of very different expression profiles of the same gene in identical tissue samples, nearly identical time course samples. Is there any way you could use the tomato pan-genome to tackle the mechanisms that control or allow for that type of expression heterogeneity? Sounds
I'm not sure. I don't think tomato is a good model because most tomatoes they are inbred line. They are highly homozygous. And if I understand correctly, I think we are talking about allele specific expression. So actually, we did some work in apple, because apple is highly heterozygous. Yeah, we have a paper in revision. I hope I can share you in the next month or so.
In what
apple.
is that?
The allele specific expression. I mean, the same genes have two different alleles.
Mm-hmm.
And then the different alleles, they have different expression pattern in the same tissue, in the same developmental course. And we found in apple, they have a lot different differential expression between different alleles of the same genes because they are from two different parents or progenitors. But I mean, in tomato, I think the Commons group, they investigate the hybrids. I mean, the F1 hybrids, and try to understand the allele specific expression, but not in one single accession. It kind of like artificially generate the hybrids, two different alleles, then check their expression pattern.
All right. So we're pushing up to 1 o'clock. I think we'll have to end it there. Apologies if we didn't answer everybody's questions. So thank you, Jim and Faye. This was great. I learned a lot here. I hope everyone out there did too. So thank you so much for doing this. Thank you everyone for joining us. And I'd also like to thank again today's sponsor, Sullivan Trading Company, providing can seamers, parts, and repair services to keep the food supply chain humming along. You can find them on the web at sullivantrading.com. And you can read more about the tomato pan-genome and many other neat stories about BTI science in our 2019 annual report, which you can find online at btiscience.org/annual report. And our annual report this year is all digital with 3 different views. There's a single page view, a 2-page view, which is most similar to seeing a hard copy, like a PDF, 2-page view, and a reading view, which is like reading an article on the web. So it's like HTML, nice and big text. And there are many links to videos and other neat interactive aspects of the online annual report. So we do hope you check that out. It's got some neat stuff in there. And I would also like to invite you to please join us for BTI's next Breaking Ground discussion series, featuring an interactive chat with BTI's president, Dr. David Stern. And that is on Wednesday, August 30th. And we'll be talking about the decadal vision, a new decadal vision. And you can please go to btiscience.org for more information about that and to register. And thank you all very much. I would just like to reiterate, BTI is an independent nonprofit research institute, and we operate in part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. And thank you so much for your interest and for your support of the Boyce Thompson Institute. And we hope you have a wonderful day and be well. All right, thanks everybody.
Thank you. Thanks.